Imagine something small enough to float on a particle of dust that holds the keys to understanding cancer, virology, and genetics. Luckily for us, such a thing exists in the form of trillions upon trillions of human lab-grown cells called HeLa.
HeLa cells have contributed to science and health in a remarkable way, reaching new frontiers that nobody thought was possible. These cells have provided scientists with a way to test vaccines and even aided with the eradication of polio as well as hundreds of other diseases. HeLa cells have allowed scientists to observe cellular processes without having to perform tests on actual human beings. For over 60 years and counting, they have played a role in many scientific and medical breakthroughs. For example, the HeLa cell line has helped with researching the effects of zero gravity on humans, leukemia, AIDS, cancer, as well as vaccines for polio and COVID-19. Now, the history of these revolutionary cells dates back to the year 1951, when a woman unknowingly had her cells harvested during treatment. 31-year-old Henrietta Lacks, a poor black tobacco farmer who lived near Baltimore, Maryland, was the mother of 5 children and had advanced cervical cancer. She went on to have surgery at Johns Hopkins Hospital in 1951, hoping to combat the immensely painful and life-altering ailment. At the time, cancer was still a mystery, much like it is today, and researchers were working tirelessly to find treatment methods, which often involved the extraction of patients' cancerous cells to observe, and Lacks was no exception to this common occurrence. Lacks had signed an operation permit, giving permission for the surgery, but without Lacks's or her family's knowledge. A cell line from her tumor was extracted. The harvested samples were simply marked HeLa, a name created by combining the first 2 letters of Henrietta's first and last name, and immediately brought to another laboratory to begin researching. Unfortunately, this small collection of cells was soon to be the last remaining piece of Henrietta Lacks, as despite the intensive medical treatment and numerous experimental trials, her cancer became aggressive and spread, leading to her untimely death in the fall of that same year. Little did she know, however, that her cells would grow to become one of the most revolutionary discoveries in medical history. Why is this? Well, previously, when scientists attempted to extract cancerous cells from other patients and reproduce them, the process failed, and the cells died before any could be duplicated. However, when scientists placed Henrietta's extracted tissues in the petri dishes, they discovered something extraordinary. These nutrient-rich petri dishes and the medium inside them allowed for the cells to grow and multiply at an exponential rate, doubling every 24 hours and never stopping, an astounding rate that set Lacks's cancer cells apart from any others previously researched.
Scientists grow human cells in the lab to study how they function, understand how diseases develop, and test new treatments without endangering patients. To make sure that they can repeat these experiments over and over and compare the results with other scientists, they need huge populations of identical cells that can duplicate themselves faithfully for years. But until 1951, all human cell lines that researchers tried to grow had died after a few days.
Something about the cells made it possible for the telomeres to continuously replenish and multiply. Telomeres, located on the chromosome, serve to protect the DNA of the cells during cellular division. The chemical compound telomerase extended these telomeres further, which in cancer cells allowed for the disease to spread. With the HeLa cells, medical researchers were able to discover that telomerase was active in cancer cells, and that by inhibiting the activation of this chemical compound, doctors may be able to slow the progression of or even treat cancer. Her cells were completely and undeniably immortal, making it possible for this telomerase research to proceed without any time or material restrictions. The cells, because of their incredible capabilities, spread rapidly around not only the United States, but the entire world. And as this occurred, the cells pioneered medical breakthroughs and revolutionized the field of medicine. HeLa cells had the ability to mimic the functions of the human body outside the body, combining for the first time medical practice with biological research. The cells were similar enough to normal human cells that scientists were able to use them in tests involving human diseases such as polio, and even in unusual experimental circumstances, such as the effect of human cells in zero gravity, or the impact of the intense heat and radiation of nuclear fission. HeLa cells were used at the time of the polio epidemic in the United States when it was nearing its peak. Dr. Jones Salk from the University of Pittsburgh was close to developing a vaccine that, if successful, would protect millions of children from the virus ravaging the country. But he needed an immense amount of cells to perform the trials necessary to test the safety and effectiveness of the vaccine. To complete the trials, Dr. Salk exposed cells to live poliovirus in samples of vaccinated blood. HeLa cells were easily infected by the poliovirus and therefore were perfect for vaccine testing and development. Since HeLa cells were easily grown and cultured in the laboratory, Salk was able to infect large amounts of HeLa cells and develop a vaccine that was viable for human use. In 1950, he began working to create an inactivated poliovirus vaccine, and by 1952, Dr. Salk had created what he considered to be a safe and effective vaccine against the poliovirus 14. The news of their immense success brought attention to the cells, causing increased demand for the cells across the world. Therefore, the Tuskegee Institute in Alabama established the HeLa Distribution Center. It produced over 20,000 tubes of HeLa cells, adding up to approximately 6 trillion in total every week. The center started selling vials of HeLa cells for $10 per to laboratories in nearly every country, including Germany. In the 1970s, Harald Zerhausen from Germany was researching the nature of human papillomavirus, or HPV for short. He used HeLa cells as the foundation of his studies, and throughout this process, he observed that not all HPV patients developed cancer, but all patients with cervical cancer were later diagnosed with HPV. In 1983, after 10 years of numerous experimental trials, he consequently established a conclusion that links the cause of cervical cancer to papillomavirus. 2 years later, the HPV vaccine was developed as one of the first anti-cancer vaccines, and he was later rewarded the Nobel Peace Prize in 2008. Without the help of the immortal cancerous cells, this may have never happened, and without a doubt, many would still be suffering from human papillomavirus. More recently, the cells have been used to study the effects of COVID-19 and to help find a vaccine for the virus. When research began with the virus, scientists soon found that this virus did not infect these HeLa cells well, something they were unfamiliar with and wanted to know more about. This curiosity led the researchers to search for the key to viral entry that appeared to be missing from the cells. Some forms of the coronavirus used a molecule called ACE2 to enter the cells. ACE2 particles surround the surface of some human cells, causing scientists to believe that coronavirus also enters cells through the same molecule. After engineering HeLa cells to display the ACE2 molecule, they observed that the novel coronavirus could then enter and infect the cells. After entry, it can use the cell's machinery to replicate and spread. This machinery was the actual RNA sequence, the foundation of the cell and a key component of protein synthesis. The virus, when it invaded the cells, appeared to alter the RNA code, which enabled the increased rate of duplication. Using HeLa cells, the researchers discovered an important aspect of how COVID-19 spreads and were successfully able to find an effective vaccine for the virus. These were only a few handfuls of the incredible discoveries and monumental breakthroughs that HeLa cells have pioneered. However, the cells have also been used in a variety of other ways. This includes studying the effects of drugs, toxins, and hormones on cancer cells, the effects of radiation in outer space, the identification and isolation of numerous other viruses such as HIV and Ebola, the functionality of these viruses and the single-cell microscopic imaging method, as well as the continuous studying of the human genome.
HeLa cells were the first to be cloned. They've traveled to outer space. Telomerase, an enzyme that helps cancer cells evade destruction by repairing their DNA, was discovered first in HeLa cells. In an interesting turn of fate, thanks to HeLa, we know that cervical cancer can be caused by a virus called HPV, and now there's a vaccine. HeLa-fueled discoveries have filled thousands of scientific papers, and that number is probably even higher than anyone knows. HeLa cells are so resilient that they can travel on almost any surface. A lab worker's hand, a piece of dust, invading cultures of other cells and taking over like weeds.
HeLa cells were one of the most remarkable discoveries in all of medical history, and they acted as catalysts that sped up the pace of biomedical and physiological research. This, therefore, led them to becoming the most widely studied cell culture in biological science and the foundation of mostly all medical developments in the last century. There is no doubt that these cells were and will continue to be a frontier in history.
And so much of the history of science and medicine was built on the backs of Black people without their knowledge. And so it's so important that Henrietta's story be told as part of that history.